name is Maverick Ratcliffe, and the title of my presentation is Identifying Symbiotic Endophytes from the Biodiverse Regions of Ecuador. The purpose of this research is to improve agricultural systems in order for them to survive the climate changes caused by global warming, and to improve their crop production to better sustain the human population of the world now and in the future. We seek to cause these changes by naturally introducing symbiotic fungal endophytes into the agricultural systems. These symbiotic endophytes are also known as symbionts, and we extract them from wild plants in biodiverse regions and areas of harsh conditions. Ecuador, which is known for its biodiversity, is a key area that we extract endophytes from. In 2019, a group went to Ecuador and extracted 72 samples and isolated them. Here are pictures taken from that trip showing you the conditions of the areas that we consider preferable. To test for beneficial effects that these symbionts can produce, we first must take the isolated sample and perform subculturing to confirm that it is pure. Next, we will make inoculating cultures. After we have enough inoculant, we can then apply it to the roots of the plants and observe them. The results from our experiments so far include the identification by using DNA sequencing, and also we have compared crop yield. Here, this is the results for the DNA sequencing, showing you positive identification in 35 of our samples. Number 36, which we hypothesize imparts drought tolerance and also improved crop production, was identified as an Adenia species. Below those results is a PCR gel. PCR stands for polymerase chain reaction, in which DNA is denatured and primers allow for a specific gene to be magnified during renurturing of DNA. In this case, we magnified the ITS region of the DNA, which is used to identify fungal symbionts. These are the results I mentioned, geared toward the crop increase produced by number 36. The NS are the non-symbiotic group, meaning that they were not inoculated. And you can see how number 36 treatments have a clear increase in crop production and outcompete the non-symbiotic. To conclude, we will continue this experiment by better identifying our samples using the biolog system. Biolog is able to identify fungal species by observing their metabolome. And because it observes their metabolome, we are also able to identify if any metabolites are what impart the drought tolerance effects or increase in crop production. We will also continue our experiments by inoculating plants with the other endophytes, but we have currently started to focus on 36.